Welcome to this video on the structure of DNA, the blueprint of life. This is your cell and molecular biology course, and I'm your course instructor in year Central. DNA, also known as deoxyribonucleic acid, is the molecule that stores genetic information in nearly all living organisms. Its discovery has revolutionized biology, medicine, and biotechnology. Now let's see the discovery of DNA. The story of DNA began in 1869 when Frederick Masher isolated a mysterious substance from the white blood cells. Later, it was identified as DNA, but it wasn't until 1953 when James Watson and Francis Crick, using Rosalind Franklin's X-ray diffraction data, they unveiled the iconic double helix structure of DNA. This discovery laid the foundation for modern genetics. So, this is how the DNA was discovered. Now, let's see the components of DNA. DNA is made up of basic building blocks called nucleotides. Each nucleotide consists of three parts: a deoxyribose sugar, a phosphate group, and a nitrogenous base. In the illustration, you can see the three parts of the nucleotide. There are four types of nitrogenous bases: adenine, thymine, cytosine, and guanine, which are abbreviated as A, T, C, and G. Now let's see the DNA double helix structure. Two strands of DNA twist around each other to form a double helix. In the illustration, you can see the two strands of DNA which is twisted around to form a double helix structure. These strands run in opposite directions, a feature called antiparallel. The bases on each strand pair specifically with each other, that is, adenine pairs with thymine and cytosine pairs with guanine. This complementary pairing is essential for DNA replication and also for its stability. Now let's see the base pairing rules. Base pairing follows strict rules due to molecular shapes and hydrogen bonding. Adenine pairs with thymine through two hydrogen bonds, while cytosine pairs with guanine using three hydrogen bonds. These specific pairings help maintain the DNA's uniform width and enable accurate copying during the cell division. So these are the base pairing rules. Now let's see the DNA backbone. The sides of the DNA ladder, that is, you can see the ladder shaped structure in the illustration, which is formed by the sugar phosphate backbone. In the illustration, you can see the deoxyribose sugar and the phosphate attached to it. The sugar of one nucleotide is connected to the phosphate group of the next by the covalent bonds known as the phosphodiester bonds. So this is the backbone of the DNA, which gives its structural integrity and also protects the genetic information within. Now let's see the DNA packaging in cells. Inside the cell, the DNA is tightly packed to fit within the nucleus. It wraps around histone proteins to form a structure called chromatin. And during the cell division, this chromatin condenses into visible chromosomes. Interestingly, each cell contains around 2 meters of DNA, but uh, we know that it's compacted efficiently within the microscopic space. In the illustration, you can see the DNA double helix, which is wrapped around the histone proteins. But as you can see, H1 histone and then the subunits of the histones together they form the nucleosome, and then you can see the nucleosome filament, which again condense to form the chromosome structure. So this is how the 2 meters long DNA is compacted into a chromosome, which is microscopic in nature. Now let's see the DNA function. DNA's primary role is to store and transmit the genetic informations. These informations are used to build proteins through a two-step process, which is transcription and translation. That is, DNA is transcribed into RNA, and then this RNA is translated into the amino acids, that is the amino acid chain, which is the protein. The DNA also ensures that genetic information is passed from parents to young ones, thus guiding in inheritance. Now let's see the real world applications. Understanding DNA has led to major breakthroughs in science. We know in forensics, DNA helps solve crimes and identify individuals. In medicine, genetic testing aids in diagnosing inherited diseases, and in biotechnology, CRISPR allows us to edit genes, offering hope for treating genetic disorders, and we can also produce genetically modified crops, and in also improving the crops which are genetically modified and beneficial ones. So DNA is the molecular basis of life. Its double helix structure, composed of nucleotides and precise base pairings, allows it to carry, replicate, and transfer genetic information. As we continue to explore and manipulate DNA, we can open doors to innovations in health, agriculture, medicine, and beyond. Thank you for watching. See you in the next video.